This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 81 for May 7th, 2010. Hi, everybody. It's Vincent Racaniello, and this is TWiV, your weekly dose of virology. With me today, from North Central Florida, is Rich Condit.
Hello, Vince.
How's it going there, Rich?
Oh, it's great. It's fantastic.
Yeah.
We have a nice day. We got, we're getting into summer here. So we got 86 degrees and a little bit humid. It would probably kill most normal people, but it's just a little bit humid for us. A few clouds, and it's real nice.
86 degrees. Well, we're just in the 70s here.
Oh, that's beautiful. Warm. That's wonderful.
But is it very humid?
A little bit. A little bit. A little bit by my standards, you know. Like I said, take somebody from, Arizona and stick them in this and they'll keel over.
I like it. Well, I'm looking forward to visiting in a couple of weeks.
We're looking forward to this too. The plans are progressing. This will be good fun.
We're going to do a TWiV from University of Florida, Gainesville in a couple of weeks.
Right.
So anybody listening who's there, and I know we have some fans there because I've gotten some emails, we're going to have an audience if you want to come and watch us do it.
Yeah, I think you got a pretty good fan base here. I expect some people to show up and watch that. I got a room reserved, so we'll have plenty of room for audience. Should be a good time.
Yeah, it'll be great. So we'll keep mentioning that every episode up until then. We're going to record that on a Tuesday, right, that week. So I think we'll do the normal Friday, if that's okay with you.
Sure.
And then we'll do it on Tuesday, release it on the weekend.
Great. Fantastic.
So today it's Rich and I because Alan is at home with his sick child, and Dixon is in Seattle promoting vertical farming.
Great.
I think he'll be back next week.
Great. But the man does travel.
He does travel. He just came back from the Canaries. He was back for one day. That was Monday. I got him to record a TWiP and give a lecture in my course, and then he was off.
Mm-hmm.
And he'll be back, I think, for next week.
Good.
And today we have an all-email episode, which will be fun because we have always great emails, and we have so many that periodically we'll just do one episode with just emails from our listeners. And before we do that, I want to bring up two items. And the first is something I had meant to mention before. I was in Edinburgh a couple of weeks ago for a meeting. And this was a meeting of the Society for General Microbiology, which is a UK microbiology society, professional society. Very nice meeting. And while I was there, two people came up to me and said, "Hey, you're from TWiV." They recognized me.
That's cool.
And so if you're listening, it was a man and a woman. I forgot their names. But I think that's great. You know, when I was going to meetings and presenting data, nobody ever came up and said, "Hey, you're the polio guy." And I think this is great that you have an international audience with the podcast. You can reach so many people. And I guess it's the fact that you're talking, people will approach you because they figure if you're going to do this, if you're going to do sort of a radio show, you must be approachable.
Right.
I guess that's what it is. But I like it. I think it's great. So if I'm somewhere in a TWiV listener, me or Rich or anyone, if you see us, come up and say hi.
Yeah. And it certainly is international. I mean, we get emails from all over the world. It's terrific.
I love that. Yeah.
Yeah.
We have, they're from all over the world. And I know that technology allows that. It's not a big deal, but I still feel it's amazing that we make this podcast and then somebody in Hong Kong or India or Africa downloads the thing and listens and then responds. I just think it's cool. You know?
The technology never ceases to amaze me. It's a wonderful time to be alive where we're so connected like this. It's wonderful.
Yeah, I like that. So that's what I wanted to mention, the Edinburgh connection. I think there are some people there listening to this podcast. So it was really great to meet you guys. Great city, except it was raining all the time.
Well, that comes with the territory.
It does. So before we move into our emails, I wanted to acknowledge the support of Drobo. We have another contest to announce today, so I want to give you the details on that. And, of course, the supporters of this episode of TWiV is Data Robotics, the makers of Drobo. And Drobo, of course, is a storage solution for your computer. And they are basically little black boxes into which you put hard drives, and then you put your data on these hard drives. And the way they're constructed, the data is duplicated over the whole hard drive array. So if any single drive fails, you just pull it out and put in a new one, and your data are restored. And they make many different models ranging from 4-drive to 5 and 8-drive models. And I have 3 of them, and I put all my data on them here at work. And I really recommend anybody in science to get one of these for your lab. And you'll really sleep easier knowing that your data are protected. And yesterday I was over at the Lipkin laboratory here at Columbia. There's a laboratory where they do a lot of virus discovery. So they're generating tons and tons of sequences on a daily basis. And there were Drobos all over the place there. And that's great. I thought that was a really good way to protect their important data. So I thought I would mention that. So if you haven't yet gotten one, you have my full endorsement. It's a wonderful product. You can even use it for stuff at home, for your movies, for your files, photos. You know, if you take a lot of pictures like Dixon Despommier does, you should put them on a Drobo, because if it's just on a hard drive, eventually it's going to fail. So we have been giving away Drobos. We're on the 3rd giveaway now. We've given away 2 in 2 different contests. Now, the 3rd contest is very straightforward. You go to bit.ly/TWiVDrobo. So it's bit.ly/TWiVDrobo, all one word. There's a questionnaire there. And just fill it out. Fill out the whole thing at the end. You put in your name and address, and you're entered in the contest. And then in 3 weeks, Drobo will randomly draw a name out, and you could win a Drobo S.
Looks simple enough.
Very simple. Just a bunch of questions.
Even I can answer these questions.
You can answer them. It doesn't take any virology knowledge, just knowledge about the things that you prefer. So they want to get a sense of who listens to podcasts and whether that's a good audience for them to advertise to. So it's easy. So check it out. You could win a Drobo. If you don't win it, you can always buy one. And, of course, you get a discount through TWiV. And if you go to drobo.com, let's see, drobo.com/ is it TWiV?
It was like TWiV Pod or something.
Okay, so it's drobo.com/TWiV. You'll go to the landing page. It says, "Welcome, TWiV listeners." They made a nice page for us. And then TWiV Pod is the promo code you type in when you want to buy one of these guys. And you can get $100 to $500 off on a Drobo. All different sizes. Really cool product, which and we're really grateful for their support. It's not going to last forever. We have a few more weeks here. But check it out. It's really a cool product. We thank Drobo for their support. Okay, let's do some email. You ready, Rich?
I'm all set.
All right. His first one is from Jesper. Jesper's from Sweden. He writes, "Dear professors, having upped my daily dose of podcasts, I stumbled upon This Week in Science. Latest show, they mentioned something that probably comes within your sphere of interest. Namely, the finding that viruses are a possible trigger for a triggering factor to gluten allergy." And he sends a link to this report from Eureka Alert. It's called "Virus Infections May Be a Contributing Factor in the Onset of Gluten Tolerance." So gluten tolerance is the celiac disease is another name for it. It's basically you have a very bad reaction to eating things that have certain grains in them. Wheat, barley, rye. You have to be careful you don't eat anything with those in them. You have a terrible intestinal inflammation.
Not a fun thing to have.
No, it's what I understand. I don't happen to have it, but I've spoken to people who do have it. So this paper is quite interesting because they suggest that, so what they did was they sequenced they tried to look for gene mutations that went along with people having gluten allergy. And the paper was published in Nature Genetics. It's called Multiple Common Variants for Celiac Disease Influencing Immune Gene Expression. So basically, they have 4,500 individuals, and they look at specific loci which they believe gene loci that they believe may be associated with the disease. So this is a SNP analysis, it's called, where you try and find variations that may correlate with the disease. It doesn't tell you that they cause them, but they give you some clues, and then you can go and investigate more later on. And so what Jesper's article is referring to is the finding that they found that two genes involved in immune detection of viral RNA seems to be associated with having this disease. Okay, so the genes are encoding proteins called TLR7 and TLR8. And TLR, of course, is toll-like receptor, which we've talked about a bit before on TWiV. And these are membrane proteins in the cell that are involved in recognizing viral RNA. So when a virus infects a cell, these proteins, among others, there are many other proteins involved in this, would recognize the viral RNA within an endosome. So a number of viruses are taken up into endosomes, and these proteins are on the inside of the endosome, and they would detect the viral RNA. And what that would do would be to trigger the immune response, first the innate immune response. You make cytokines, interferons, try and clear the infection, and if that doesn't work, eventually you get the adaptive response. So they think that this may be involved in the inflammation in the intestine, in gluten allergy.
So if this is supposed to imply that there might be a virus or viruses involved in the allergy, it seems to me a pretty indirect implication. It's basically what they certainly identify is that there are things wonky in the immune system, and this being, I guess, essentially an autoimmune disorder, that certainly makes sense. But it seems to me that it's a fair leap from this to actually implicate viruses directly in this, or am I looking at this wrong?
No, I think that's right. I mean, there's certainly no specific virus implicated. I mean, it could be that any time these people are infected with a virus in the intestine that it triggers this, in conjunction with, eating the wrong kinds of foods.
Mm-hmm. At the same time, the notion that viruses, the implication of viruses in autoimmunity is not a new idea, and it's a very interesting idea. The idea that some viral antigen may trigger a cross-reactivity in the immune response with some self-antigen.
Sure, so, absolutely.
And that kind of thing could be going on here as well.
Some other ideas I had were, we have lots of viruses in our gut normally, and maybe in these individuals there's a mutation in the TLR7, TLR8 gene that makes them aberrantly react to these viruses.
Good point. Yes,
especially when there's some gluten present. Or maybe these sensors just fire aberrantly without even having a ligand around. Right? So it could be any of those things. So yeah, there's no specific virus, and it's not even clear if a virus is involved in this. But it's an interesting finding, and now you can do experiments to look at this further.
So have you listened to this This Week in Science? I have not.
Yeah, I have listened to it now and then. It's, as he says here. Let me read what he says. He first summarizes the findings, saying, I presume this may open up a huge field of both treatments and prevention. So that might be true. As for This Week in Science, it's a general science news pick kind of show with an attitude, not as in-depth subject-focused as TWiV, and no doubt satisfying a different need, but anything helping the scientific project is worth trying. I think Vincent may appreciate their music selection, but that's mere speculation. So I have listened to This Week in Science now and then. It's a popular science kind of show. Mm-hmm. They cover a lot of topics, kind of not very deeply. Mm-hmm. So it's different from what we do. But as he says, if it makes people learn about science...
That's great.
I think it's great.
Yeah.
I think the music's okay, Jesper. Oh, great. I like our music better.
I'll have to listen to it, see if I can
Anyway, thanks for that,
start.
Jesper.
Yeah, it's great.
Hey, Rich, why don't we alternate on reading these? Do you
Sure.
mind? Take the
Sure,
next one.
not at all. Jason writes, I was listening to the most recent episode of TWiV, episode 72. So these go back a ways. And I was delighted to hear you discuss RNA silencing in viruses. It happens to be what I've devoted the last 6 years or so of my life to, and it's a topic that I'm fairly familiar with, and I just wanted to shed a little light on some things. First, a brief background on myself. I defended my dissertation at the end of last May, graduating from NC State University, where I performed research on a fairly obscure plant virus, red clover necrotic mosaic virus. My research surrounded the role of plant RNA silencing as a host defense mechanism, and of course, how the virus might circumvent this defense. Towards the end of my research, I knew I wanted to transition out of plants into animal virology, so I applied to a variety of labs and ended up getting a postdoc position at Duke with Brian Cullen. It's been an interesting transition, and I've absolutely loved listening to your podcasts, which I think do a great job of providing digestible information for everyone from the common layperson to PhDs like myself. Anyway, at one point in the past episode, you made a comment that all vertebrate viruses are surely chopped up into siRNAs. Rich wasn't so sure about that, and he is definitely correct. That's a first. I, I won't say that it is a completely settled subject, but at this point it does not appear that RNA silencing is a general feature of viral defense in vertebrates, although some exceptions may exist. Certainly there is an interplay between small RNAs and viruses, but a generalized antiviral link is pretty tenuous at this point. You mentioned how it would be useful to do some deep sequencing on infected cells to see if you could detect virally derived siRNAs. Our lab has done that for a number of herpes viruses, and along with other labs, have found that herpes viruses actually encode their own microRNAs. In fact, EBV encodes 25 microRNAs, while KSHV, HSV1, HSV2, these are all herpes viruses, encode multiple microRNAs as well. I can't recall the number off the top of my head. A much broader study was recently published by Andy Fire and colleagues in PLOS Pathogens, which you might find interesting, and he gives the link. Keep up the good work. So yeah, after that show, I checked on this myself because, as he points out, I wasn't so sure, and I checked with some of my colleagues who are better up to date in this field than I am, and they basically came up with the same thing, that no, it's not. It's at best controversial whether there are viral SI or interfering RNAs in mammalian cells. In this paper that he's sent us does, what does it do? Deep sequencing on, I haven't looked at this recently.
Yeah, they infect cells with a variety of different viruses, and
right.
And they look at the small RNAs, so they purify small RNAs and just sequence them.
And they do find a few.
They do. They find quite a number, actually. They find, for all the viruses, hepatitis C, polio, dengue, VSV, West Nile, they find small RNAs, which they call virus-specific RNAs, which are mostly seem to be derived from the genome, although they don't actually know the origin of all of them. And so they say some of them are really vanishingly rare and others are abundant.
Mm-hmm.
So the idea is that it could be that
the
cell is chopping up the viral genome, and making small interfering RNAs, which would then be used to guide degradation of the virus RNA.
That's the implication, yes.
Yes, but they don't know.
It's going to have to be established.
Yeah, so in the discussion they say this could be, but there are lots of other possibilities. But so what we know from this is that they exist. They occur in virus-infected cells, as Jason has told us.
So the other thing that he points out here is a topic that we really ought to take up in some detail on another TWiV, and that is the microRNAs encoded by these herpes viruses. As a matter of fact, you suggest here we ought to get Brian Cullen to at least listen to TWiV, if not have him on the show, because he was here not long ago and gave a seminar about the microRNAs and herpes viruses. And actually, we have a faculty member here who was one of the co-discoverers of microRNAs in herpes viruses. So I've heard quite a bit about this, and it's a fascinating topic. And Brian's perspective on it was very interesting as well. He had a lot to say about why you might find these microRNAs in some viruses and not others. And there's a lot of investigation going into what effect they have on the virus life cycle. It's a very interesting topic.
Yeah, I heard Brian at the Edinburgh meeting.
Oh, okay.
He talked about this. I would love to have him on TWiV. So Jason, tell Brian to listen up and convince him to come on the show. How's that?
Absolutely.
He'll probably listen to you. Great. Thank you, Jason. All right,
and good luck.
Good luck. Great stuff. So I think this is a great example of how we touch everybody with TWiV. Mm-hmm. This is a PhD person, postdoc, and, he thinks it's great. And so it's just pitched at the right level. All right, the next two emails have to do with the mumps outbreak that we talked about a couple of times in the past. And you, Rich, had a friend or a former student, I think, at CDC who was involved in the exchange. Mm-hmm. And so Ewout from the Netherlands, we had answered one of his questions, and he says, "Thanks for answering my mumps question. Great fun and honor to be cycling back from work and suddenly hearing my email on the iPhone."
He does the same thing I do. Listen to TWiV on his iPhone while biking to work. That's great.
Me and my colleagues would like to read the CDC response mail with the references named in the podcast. Would you be so kind to forward it to me if possible? Hope you are able to continue this great podcast for a long while. I will advise more colleagues to listen to you. So yes, your friend at CDC had sent a really nice response, which we talked about. So I sent it to Ewout. So he replied, "Thanks. We, the National Institute for Public Health, are planning to go deeper into the mumps issue, since vaccination-related diseases, measles, mumps, etc., are an ongoing issue because of areas with low vaccination coverage due to Protestant Reformed religion. Genotype G suddenly seems to hit vaccinated non-religious students. I also contacted Israel a few weeks ago, thanks to your former TWiV about their outbreak. TWiV is more than just an interesting podcast. It enhances science."
That's very interesting. I mean, it's having an impact actually in spreading information and putting scientists in touch with each other. That's great.
So that's why I wanted to highlight these, because we could get scientists together who wouldn't normally think of something, and they hear it on TWiV. We can connect them, and certainly other ways that they can do that. But it's a good side effect. There you go. Your turn, Rich.
Okay. Bree writes, and this is from the, Indiana University in Pennsylvania.
Right.
The IUP virology class. "Hello again. I just wanted to say thank you once more for taking the time to answer my question on your podcast. It's very exciting for me and the other members of my class to be able to have our questions answered after listening to your podcasts. And to answer the question you asked during the last podcast, I have mixed feelings about podcasts as a learning tool. For someone like me, who only has a very basic grasp on the principles of virology, understanding everything that is being discussed by people who are very advanced in the field can be difficult. However, when I do understand the subject matter, and hopefully I will after completing this course, I find that it's relatively easy to follow and understand what's being said. I also find that listening to what someone other than my professor has to say about the topic being discussed is not only refreshing, but provides me with more than one viewpoint from which to understand and look at certain topics and new material. Again, I greatly appreciate all of you taking the time to read and answer my question, and I look forward to hearing more questions answered in future podcasts. Thank you, Bree." Well, that's nice. The other nice thing about the podcast, and I do this myself, just like a recorded lecture or something like that, is that you can back up and listen to bits again, and because your mind wanders or you don't quite get it or whatever, and so you have it right there, and you can listen to bits over and over again and hopefully get it.
Yeah. This is a theme that you'll hear in many emails, that a lot of people don't understand it, or parts of it at least, but they say they listen anyway, and eventually some of it rubs off and they get it. And it's a good thing about a weekly podcast. You can hear things multiple
You
times.
get it reinforced because
Yeah.
you hear it multiple times. Yeah.
Jim writes, the Canadian Broadcasting Corp podcasts are my only connection with Canadian activities. Too bad we don't hear more about our neighbors. I'd not heard about the Summit Awards until this podcast, which he links to. Not much about viruses, but listeners to TWiV might still be interested from the standpoint of career development and jobs. So it's a 46-minute program, Science at the Summit, and it basically is two leading Canadian medical researchers, Benjamin Neal and John Wallace, discuss what it takes to rise through the ranks and ultimately arrive at the lofty pinnacle where true scientific advancements are made. They got some prizes, the Premier's Summit Awards. Wow, the lofty pinnacle. I'm not there. Are you?
No way. I'm just hanging out.
Oh, we're just doing experiments, which is
I'm
really
just,
what you need to do.
I purified a whole slug of virus this week, and I was real happy about that. That was good fun.
Well, thanks, Jim. We'll pass on that link in case you want to find out what's going on in Canada. And he continues, in case I've not said so, all the TWiV podcasts have excellent sound quality and levels. Well, thank you. I've heard several references to online videos of lectures from places like MIT at academicearth.org. After viewing a dozen or so on chemistry, math, biology, computer science, and physics, I must say how much more enjoyable the TWiV podcasts are. Lectures do not appear to have changed from the last I attended 25 years ago. I expected to see more. In several, there wasn't even any chalkboard work, just a lecturer talking. Just a comment. Well, the podcast is an informal way to learn. That's what's neat about it. It's not structured. You know, you don't have to get everything because we'll come back to it at some point.
Right.
I think that's possibly why it does work. Of course, in a formal setting, in a course, it may not work as well because it's not as structured, but...
Yeah, well, it's interesting, and the best lecture courses are, in fact, lecture courses where there's a lot of discussion that goes on during a lecture.
Yeah.
In which case, it takes on more of the flavor of a podcast. I think it's a lot more interesting and stimulating. It's easier to pay attention if there's a couple of people talking, asking each other questions and that kind of stuff.
Yeah, instead of a monotone,
Mm-hmm.
one person just talking, you have a couple of people, different tones of voice, different topics.
Right.
I like it. And that's why you're right. A discussion class is always much better when the class participates. I always like it when my virology class talks. I think
How many people in your virology class?
I have 44.
And did you teach the whole semester?
All but 4 lectures, yeah.
So how many, you taught like 40 lectures or something like that?
No, it was, the whole course was 26 lectures. It was twice a week. Yeah.
Twice a week.
Yeah, and we're having our final exam on Monday.
And these were undergraduates?
Undergrads, yeah.
All right. Boy, they got a treat. That's great.
I had a good time. And
Good.
in fact, one of the assignments was to send in a question to TWiV.
Ah, okay.
And I got a bunch of them, and we'll be hitting those over the summer.
So is this a new course?
Yeah, it's a brand new course I started this year.
Are they science majors or non-science majors? What's
It's a
the deal?
mix. I think most of them are science majors, a mix of a few others and non-science majors, a few older people who are going back to school.
Had there not been a course like this before?
Not since the 1980s.
I'll be darned! Wow,
And I
that's
figured
great.
it was a good opportunity. So
Absolutely.
it's down at the Morningside campus. I don't know if you know, the medical school and the other campus are separate here in New York.
No,
Columbia.
New York is, what is it? Vonnegut called it Skyscraper National Park, and
Right.
that's kind of what it is to me.
So we go down to take the subway. It's 40 blocks and teach it down there. But I love
Great.
it. It's great. It's a real campus down there. and it's
The
a lot of
older
fun.
students must be interesting. People coming back again.
They're all very interesting. I've enjoyed it a lot.
Okay, good.
One day I
Great.
came out of the course and Salman Rushdie was there on the sidewalk getting into
Oh.
a taxi. So that's New York, Actually, he's an adjunct. He does some teaching there. All right, your turn. Yeah.
Mike writes, I was just listening to the show after downloading every episode, and I'm really enjoying it. Oh, I've just started listening to the show after downloading every episode, and I'm really enjoying it. I have no science background, but I'm a geek and enjoy listening to Futures in Biotech. And so as soon as I heard about TWiV and TWIP, I knew I'd have to check the shows out. A worthy find indeed. I shall be recommending it to other science-minded friends. Keep up the good work. Well, that's nice.
Good.
Thank
Every episode, huh?
Yeah.
I
I did that at one point. After starting up on it, I went back. I had to catch up on, I don't know, 1 through 25 or something like that. That's a task.
It's a lot. Yeah.
You can do it.
Richard writes, thanks for posing the question. It has proved interesting to research and to think about. Oh, he's referring to our Drobo contest. Can you engineer influenza to replicate in bacteria? I have a pick for you. This is a web page that provides information for experimenting with phage at home. It gives information and experiments that require little or no equipment to be purchased. Yeah, this is a great web page I wanted to pass on. Indiana Biolab Bacteriophage Index. So these are experiments you could do at home. It's great.
Phage are wonderful for this kind of stuff, because they really are. It's all, toothpicks and a few petri dishes. You can do this. I was looking through this. There were quite a few links that are broken or something. So whoever's running this guy, if you're listening, get it back together because this is interesting.
Oh yeah, there are a bunch that are broken. In fact, maybe most of them are broken.
I think it's not real new, but you got a place where you can write private email messages to the guy who's running it. And so if you're interested, maybe we can get him to update it.
Yeah. All right, we'll do that. Speaking of phages, there's a fellow in Pittsburgh, Graham Hatfull. Ever hear of him?
Quite. So I had one of my PhD students about, some of the listeners might be interested in this, one of my PhD students about halfway through his run in my lab, came to the conclusion that he really didn't want to pursue an academic career in a research-intensive university. He'd like to teach in a undergraduate emphasis college, and his role model was his alma mater, James Madison University, which is basically a 4-year, it's a university, but mostly undergraduate programs where the faculty are expected to maybe run a small research program for undergraduates. And I said, well, Steve, if that's what you want to do, you can't work on a pathogen. You got to do something that's safe. You can't have radioisotopes. You can't have animals. Okay. So if you're interested in viruses, do phage. And I introduced him to Roger Hendricks, who is part of this Pittsburgh Phage Institute. And there's 3 guys involved, Hendricks and Graham Hatfull. And I'm sorry, but I forget the third person. And Steve wound up doing a postdoc with Hatfull. And he had a wonderful time. They do a lot of phage discovery. They interact with a lot of high schools and undergraduates. And Steve wound up getting the job at his alma mater. So he's faculty there now. And he wrote also an area grant, one of these small grants that funds laboratories in 4-year colleges to try and stimulate research there. And he wound up getting a grant. So he runs a phage discovery laboratory in this place, having trained through the Pittsburgh Phage Institute. Wonderful, wonderful story.
Well, Graham Hatfull was also in Edinburgh. He gave a talk.
Okay, great.
And he was at the session I spoke at, and I talked about TWiV and stuff, and he was in the audience, and he said, do you guys talk about bacteriophages? They are viruses too. And I said, of course. He said, because I want to make sure the message gets out. And I said, Graham, come on TWiV and get your message out.
Absolutely.
So we're trying to work out a date. He's going to come.
Okay, good. Excellent.
Yeah, he's got a great program. As you said, they have the students go out in their backyard and discover phages. It's called No Phage Left Behind.
Yeah, and there's a phage hunters thing as well. As I said, they've got this. It's wonderful because they've got a program that gets high school students into the university labs. And you can get a student out who goes out and discovers a phage and brings it into the laboratory and gets a genome sequence, right?
Yeah, that's right. They sequence them. They send them out for sequencing.
As a bonus, they get to name phage, right?
It's very cool.
So
we
really
have to have Graham on and
absolutely
tell you guys about it. Yeah. All right.
Let me see.
Thanks,
my turn, right?
Thanks, Richard. Yeah.
Yeah. Thomas writes, hello, love the podcast. Wish everyone was as enthusiastic about what they do as you guys are. I have some questions and clarification. 1. On TWiV 65, a pick of the week was Rebecca Skloot's book, The Immortal Life of Henrietta Lacks. The HeLa cells are infected with HPV-18, but I cannot find any discussion of what makes these cells immortal. Certainly, experienced virologists such as yourself can help in my quest for an answer to this question. So let's do these one at a time.
Sure.
So this is actually another thing we ought to do a podcast on. It's one of my favorite subjects. The papillomaviruses, like many DNA-containing viruses, contain genes that are there for the purpose of stimulating the host DNA replication machinery, because that has the effect of, since the viruses are parasitizing the same machinery, of boosting the replication of the virus. So if you need host DNA replication machinery to replicate your own genome, it makes sense to go in and stimulate that machinery to happen. Well, if you stimulate host DNA replication machinery, you're going to stimulate cell cycling. That's potentially cancer. And then there's this little dance that goes on between the cell and the virus, where the cell says, no, you're not going to do that. And the virus says, oh, yes, I am. Blah, blah, blah. But these genes in the virus that stimulate the host cell to make DNA and ultimately cycle are called oncogenes, because they promote cancer. And in the human papillomaviruses, they don't ordinarily integrate into the genome, shall we say, deliberately. It's not an obligatory part of their cell cycle, but it can happen accidentally. And there are two outcomes of that. One, if it happens in a certain area of the papillomavirus genome, it blows off a regulatory gene so that those oncogenes now are expressed at a higher than normal level. And two, it integrates those oncogenes. So now, because the HPV DNA is there in the genome in a certain configuration, you have these oncogenes, the genes that stimulate DNA replication in the host cell, turned on at higher than normal levels and permanently. And the result is cancer. Right? Now, it doesn't happen. There are probably cofactors involved, because not everybody that's infected with these viruses gets this. It's only a very, very small percentage, but that's the fundamental basis.
So those viral genes are called E6 and E7, right?
Right.
So they mess with the cell cycle, basically, and make them keep dividing. The other thing I never understood, so these cells that are transformed and immortal because of these oncogenes have to find a way to keep the ends of their chromosomes intact. And in those cells, the telomerase, which does that, is activated constitutively. I don't know how that happens.
I don't either. Yeah. I have no insight into that.
Yeah, I've asked some people, and I don't think it's known, but that's the other requirement, of course, for the cells to be immortal. The telomerase has to be active. But I think you're right. We need to get a HPV person.
Peter Halley. Got to get Peter Halley on the show. He'd be terrific.
All right, we'll do that. I actually sat next to him at dinner last year, and he was very curious about the podcast. So he's been primed.
Okay, good.
We'll get him on.
Let's see. 2. There are 2 current rotavirus vaccines and 1 previous. Here is a history and a question. You can omit the history if it gets too long. No, this is interesting. RotaShield FDA was approved in 1998, a lyophilized tetravalent rhesus human reassortant rotavirus. It is postulated that because it is rhesus and not bovine-based, that the closer genetic similarity allowed it to replicate in the intestine and lead to intussusception. So it's important for listeners to understand. So the rotaviruses cause gastroenteritis. They are reoviruses. They have a double-stranded RNA genome that is segmented. Okay, so it comes in like 10 segments, I guess, depending on the strain. So in a way similar to flu, it's very susceptible to reassortment. So you can infect cells with two different viruses and get out a virus that has a combination of segments from each of the two viruses. And these reassortant vaccines are very clever. I don't understand them completely. I don't understand who's got what segments or that kind of stuff, but they are reassortant viruses from different sources. In this case, monkey and human. Later on, we're going to get one that's cow and human. And I guess the idea is they can still replicate, but since they have genes from another species, they are attenuated. And what he's saying here is that intussusception, I guess, is not an uncommon consequence of rotavirus infection anyway. And what they're saying here is that since this vaccine may be given that it has segments from monkeys and humans, may be close enough to the human virus so that it could lead to intussusception, which is why that vaccine had trouble.
Now, just a coda there. That vaccine was developed in part by our friend Harry Greenberg, who's a gastroenterologist at Stanford. Mm-hmm. He was here a few weeks ago. He said it's being clinically trialed in Africa, that vaccine, because the other vaccines don't work in Africa. They don't seem to confer sufficient immunity.
Interesting.
And he is of the feeling that it was never really proven that this vaccine actually caused an increased level of intussusception.
Yes, I've heard that. As a matter of fact, I think I've heard that directly from Harry. I think the statistics were marginal. Okay, right. It was a small effect, if any.
We'll get Harry on sometime to talk about. He's actually an interesting guy.
Yeah.
But intussusception is when the small intestine goes into the large intestine there where the two join around the appendix area. The small goes up into the big one. And Harry said nowadays all you do is put some air in your intestine and it pops back out again. But
I'll be darned.
In the old days they used to have to do surgery to fix it, but they learned that wasn't necessary.
Oh, that's great.
All right.
Rotatech. This is the next rotavirus vaccine, is a pentavalent human bovine reassortant rotavirus vaccine. It has been wonderful for us primary care pediatricians. That's what this guy is. Reduced rotavirus-related gastroenteritis hospitalizations by over 98%. So that's good. The intussusception correlation has not been reported, but this vaccine is given to infants less than 6 months old, and if you're 12 weeks old, you're too old to start it. These restrictions were put into place because intussusception after RotaShield correlated with increasing age of the first vaccine and increasing age when getting any dose. Hard to imagine that at 12 weeks of age you are too old for vaccine. No jokes about the experienced virologists in this podcast at this point. But that is what we have to tell the patients. Rotarix, just today the CDC reported, and we've talked about this before, that there is porcine circovirus 1, PCV1, present in Rotarix and has been withdrawn from the market. It is not swine origin, just seen in pigs. They do not know if there is an intact virus present or just DNA segments. I think the ultimate sequencing said there was virus there, right?
That's right.
Because it was protected from DNAs.
Yep.
Or whether it causes human disease. From what I can tell, no evidence that it causes human disease. They also do not report how the virus was detected. We've solved that. It was sequencing. Can you dig into this and enlighten the masses? Let the second coming of swine flu begin in the media. They get things wrong a good part of the time so that we can get to work undoing their mistakes. So we covered the PCV1 thing. It is virus. They discovered it by sequencing. There's no evidence that it causes any problems in humans. And I guess that vaccine has still been withdrawn, but they're looking into it.
This is actually, even though this email is about a month old, it's very timely because just yesterday, the FDA announced that they have found porcine circovirus DNA in Rotatech.
Oh, no kidding.
Yeah, the Merck vaccine, which you just mentioned.
But now, didn't they do Rotatech as part of the study?
They did. So the original study, which was published in the Journal of Virology from Eric Delwart and his colleagues, they had looked at the Merck vaccine and did not find porcine circovirus DNA
So it must be there at a much lower level or something. Is that right?
Yeah, so Merck provided this information to the FDA. They said it's lower level than found in Rotarix. So they are meeting today, actually, to decide what to do, because now both vaccines have this, and they can't hold one back and not the other, I suppose.
Right.
So they have to either do both or none. So they have
Well,
to make
it
a
presents
decision.
a really interesting thing. First of all, as we've said before, the more capability we have to discover these sorts of things, the more we're going to find that these other agents or other things are present. And then the question becomes, what do you do with the information? Man, if you withdraw both of these vaccines from the market, now you're going to have the gastroenteritis issues again. Which do you want? I don't know. That's a tough decision, you know.
Yep.
But there is no evidence that the PCV1 does anything, and all the trials were done with it. So, and you know, you're probably eating PCV1 all the time. So
That's a tough one because it does not seem to be associated with any problems, but who's to say in 20 years it would be, right?
Right.
I would just as soon get it out, but to do that requires a whole new vaccine to be made.
Right.
It would take at least a year probably.
Right. So to finish off his email, here's a good tagline for those who want answers now that we don't know. Slow progress is better than no progress. Have a great day. That's from Thomas, who is a pediatrician. Great.
Wisconsin, yeah, great. Glad to hear you guys are listening. All right, the next one's from Joel. TWiV episode 72 was great. I had a few quick comments. One, the original high-profile paper describing CRISPR as an adaptive type immune response in bacteria was from a research group at a dairy company. Apparently, there are some common bacteriophage that are the scourge of cheese and or yogurt production. I found it interesting that these phage are such a problem that the company funded basic research like this.
That is actually very interesting. Okay. And I can imagine, I worked on phage as a graduate student, and we had times when there was just so much phage in the air in the laboratory that you couldn't grow any bacteria. And they're just deadly. They come through and just wipe everything out. I can imagine that in the dairy process where you have bacteria that are an important part of producing your product, that it could be a serious problem.
Wow, so this is a 2007 science paper from Danisco USA Incorporated, which is in Wisconsin, and they found these CRISPR sequences, which you remember, there are sequences in bacteria which are derived from phages or plasmids that come in the bacteria. It's kind of like an immune memory system. Talked about this in 72. All right, that's good to know.
Yeah, and this is real serious science that this dairy company is doing.
It's great.
Good stuff.
It's great. Commended to them for letting it go forward. Number 2, while it is well documented that plants and insects use RNAi, siRNA mediated, as a defense mechanism against viruses, RNAi, either miRNA or siRNA mediated, as a defense against viruses in animals with more complex immune systems, has been contentious. An interesting exception is in germ cells, a novel class of small RNAs called Pee-wee interacting RNAs, piRNAs, are highly expressed in germ cells and are thought to defend these cells against the replication and reintegration of endogenous retroviruses. Stringent defense against the replication of endogenous retroviruses is important to prevent ever-expanding genomic junk from being passed on to future generations.
Interesting.
So again, visiting the RNAi or siRNA question that we just talked about. I've heard of these piRNAs, but maybe we should talk about them at some point.
Right. There's a lot of these small RNAs running around, and I'm starting to get confused.
Yes.
Yep. We probably need a program to sort them out.
If we are, then our listeners must be, except for the specialists who know a lot about them.
Right.
Number 3, if you're interested in discussing archaeal viruses, I recommend having Dr. Mark Young from Montana State University as a guest on your show. His group has been spending years finding viruses in what amounts to boiling acid in the hot springs of Yellowstone Park. Wow. Yeah, do you know Young?
No, I do not.
Yeah, I've heard of him. I think I've seen him talk at ASV. And of course,
So that would be great. That comes under the heading of Phage. We'll get him and Hatful on, and we'll have a regular Phage show. It'd be good.
so Dr. Young is in Bozeman, which is
That's
where
where I asked. Oh, hey,
we're going to be there this summer.
yeah.
Yeah.
We'll undoubtedly be able to look him up there. That's great.
Yeah, he should be at the meeting.
Sure.
Yeah. So we could say, hey, listen up and come in the program. So this writer, Joel, received his PhD studying rotaviruses and has now moved on to a postdoc on leishmania. So I enjoy both TWiV and TWiP. Keep up the good work. P.S. Dixon, any chance you'll be at the Woods Hole immunoparasitology meeting in a month? No chance, Joel. Dixon doesn't do that anymore. He goes to vertical farm meetings, unfortunately. Well, that's fortunately for the world.
Right.
Gonna make food. Thanks, Joel.
So Henrik writes, I really enjoy your show, and I'm slowly starting to get the hang of basic virology. I like the way you revisit topics from different angles. It helps to hear things again in a different way so it doesn't become boring. And don't forget the 101 stuff. You haven't done a 101 in a long time, I don't think.
Nope, we haven't. Yep. Since I've been teaching, I didn't.
True. Well, actually, you've posted a lot of your lectures, have you not?
I have, but as we said, the podcast is different, right?
Yeah.
Yeah.
So don't forget the 101 stuff. Even if I'd rather learn about the sophisticated topics, the devil is usually in the details, so understanding the basics is vital for really following your trains of thought. I just watched a nice talk on Fora.tv about an expedition to New Guinea. You may find it interesting that they did a lot of virus sampling in birds there. Keep up the great work. Now, I actually didn't look at these links. Vince, did you?
Yeah, there are movies one of them is a movie on this New Guinea expedition. And talk about how they're looking for viruses in the birds there. It's very cool. I don't know anything about that. And then the second is a web page just describing the expedition. It's got great pictures of the boats and the area and a little description of what they did.
Cool.
I'd like to go there and sample viruses myself. Looks like a lot of fun.
Right.
So I will post these, of course, and have pictures of these guys standing on the shore, looking out in binoculars, standing on boats, then catching birds and sampling them. A lot of human waste there, so they want to see.
My goodness, that does look fun, doesn't it?
They're sampling DNA and RNA to look for viruses and other diseases that may correlate with bird abundance and distribution. This looks just great.
My goodness.
And these birds are cool. They have a lot of birds that they sample and then release, so check that out.
I'm heavy into boats, and they got a lot of nice pictures of these boats, too. Very cool.
All right, thanks, Henrik.
Right, yeah.
Yeah, we'll get back to 101 now that my course is over. We'll do that. This one's from Dan, who writes, your recent episodes have again discussed aspects of H1N1 and influenza. Given advances in gene sequencing, deep techniques, or whatever, it seems that if these techniques become economical, swine populations or birds could be monitored for new influenza strains on a regular basis. Once a new, say, novel infection is discovered in the swine before the virus becomes zoonotic, did I say that right?
You did?
Zoonotic. He said zoonoic. It's zoonotic.
Okay.
Or transmitted to humans, an inactive vaccine could quickly be made from the swine virus through, say, for instance, plant or insect techniques common by that time that could be administered to humans along with the annual flu shot. I don't think the vaccine would be harmful to humans, an active vaccine, but it could be an intelligent shot in the dark to head off a potential nasty pandemic via cross-reactivity. Did I state this right? Is this idea science fiction, or does it have potential? Until a true vaccine for all flu types becomes reality, it seems this could actually be a way to protect our growing population from a devastating infection with high virulence. Remember, when you end your podcast with another TWiV is viral, I am the one who was just infected. Yeah, you're right. Yours sincerely, Dan, the meteorologist in Colorado. And then he says, P.S. give my threes to Alan. He will understand.
Do you understand that?
No, but Alan's not here. I'm sorry. I thought I queued this up for Alan, and he's not here. So,
well,
we'll send it to him.
Hopefully Alan will, at least have a look at this, and so we can solve that mystery. So I think that one of the things that we discussed is that perhaps closer monitoring of pigs, at least, um, for uh, the maintenance of potentially pathogenic influenza strains is something to think about, at least. Whether you could, I don't think we know enough to anticipate what's going to grow in humans, right? And there's probably all kinds of flu in pigs. So making an appropriate selection of something to make a vaccine against would be very difficult. Basically, the real heads up is what viruses wind up actually circulating in people and causing diseases.
Exactly. No, that's it. I don't think you can immunize against something that's not yet in people, right?
Right.
So
You'd be pretty busy.
and there's so many candidates in pigs that you wouldn't know which one. You cannot.
Nevertheless, keeping your eye on the pigs is not a bad idea.
No, I think surveillance and deep sequencing, absolutely, to see what's there so that you have a head start to then when you see something in people, you say, we have this virus in the freezer, something like that. But I don't think you can give people a vaccine for something that isn't yet in them. Just think, I mean, people didn't even want the pandemic vaccine of last year because they said it was too mild, right? So, but I agree, Dan, surveillance is key.
And we'll give your 3s to Alan. And he must understand, I guess.
Yeah.
Okay. Anthony writes, Dear heroes, iterations of flattery and humility. A question. Does an individual who is infected with HSV, these are great questions, by the way. Does an individual who is infected with HSV1 and who chronically develops lesions become newly infected with each outbreak, i.e., if retroviruses insert their DNA copy with impunity, would not each outbreak produce new virus to peruse the body and infect other cells, or even the same cell twice? It is my understanding that HSV1 infects neurons in the face and neck. I'm going to stop there because he's coming on to a different question. Let's deal with the first one first. The recurrent, first of all, the implication here is that he's kind of equating retroviruses with HSV1, and it's important to understand that the mechanism of latency in those two cases is different. There's a lot of similarities. There are similarities from the point of view that the virus genome hangs around permanently. In the case of retroviruses, it's actually integrated. In the case of the herpes viruses, it's not integrated. It exists as what we call an episome, a circular double-stranded DNA molecule that's in the nucleus, but not covalently hooked up to the DNA. Also, in the case of retroviruses, there's evidence that, well, at least on the organismal level, there's a little bit of virus leaking through all the time. So they're not totally latent, whereas in HSV1, although this is kind of a squishy concept, the latency is much closer to what I would call a true latency, where the virus is really shut down and the genome is quiescent for a long period of time. And to answer the direct question, if, for example, you're dealing with cold sores, which is HSV1 that's harbored in your ganglia of your head and neck, as he says correctly here, each time you get a recurrent cold sore, that is the latent virus being reactivated and coming back to the same spot. It is not a new infection. And though I could suppose, both in the case of retroviruses and HSV1, since there's virus there, active virus there is the potential that could spread and infect other cells in the body. Would you agree with that, Vince?
Yeah, absolutely.
Okay.
Yep.
The same cell twice, well, in an acute infection, the same cell's cooked anyway, so I don't know. And in the latent infection with herpes, the latently infected cells, they apparently survive, the neurons apparently survive and reactivate many different times. So the getting infected twice as they're infected once and produce virus on multiple different occasions, I would say.
There may even be some superinfection exclusion, right?
I suppose there could be, yeah.
As there is with the pox viruses.
So, do viruses newly produced during an outbreak infect other nearby neurons? Do they insert many copies into the same infection-susceptible cell? My guess is no, because this would eventually be horribly detrimental to the host. If my guess is inaccurate, how many outbreaks must one go through before a secondary infection induces cancer or apoptosis in these rarely regenerated neurons? Well, in the case of herpes, I think there can be multiple genomes, but not a whole ton in a given neuron. As I say, they aren't integrated. They're just harbored there in a quiescent state. And the question of whether or not an outbreak can affect nearby neurons, we had two different answers to this. You asked the question to Saul Silverstein, right?
Right.
And you go ahead and read his answer.
He said, given that not all neurons that compose a dorsal root ganglion are infected, it is entirely conceivable and highly probable that other uninfected neurons could be infected during the course of a recurrence. After all, the virus spreads laterally, although because of circulating antibody, that spread is rather limited.
And I asked the same question of my colleague David Bloom, and he said, when HSV productively replicates, including during recurrence within neurons, it is not efficiently, if at all, spread to adjacent neuron cell bodies in the ganglia. There's some evidence that virus replicating at the skin during a recurrence can infect new nerve termini at the periphery. But in general, you do not see a widening zone of recurrences, indicating that this process is probably not very efficient. During recurrence, you do have pre-existing antibody and cell-mediated immunity that controls things somewhat, even in people that experience recurrences. So I think one of the things to say about this is that it is a controversial topic. Okay, the bottom line probably isn't in on this, and it's a very good question. I'm fascinated by the fact that the neurons that harbor these things and are reactivated continuously seem to survive.
Mm-hmm. Well, that's the hallmark of latency, right?
Right.
The lytic cycle has to be regulated so the cell isn't killed. And there are many ways to do that. You know, you dampen replication, you turn off proteins that would otherwise kill the cell, etc. But otherwise, it's a lytic infection and the cell's dead, and that's it. But obviously, it's been modulated in this case.
So did we lose a part of this question? You had a question about lysine.
It was another person. I pasted the wrong answer here.
Okay, fine. Another question. How can a lowly research technician get money to study a brilliant idea he or she had? Then how can said technician get the IP for if his or her brilliant idea really works?
Can't.
Write a grant.
If you're working at a university, you sign away your IP.
Right.
It belongs to the university.
Start a company.
But,
get some fun. Get a grant.
You know, the PI gets a cut of the any money that is ever made off of an idea, and
depending on the university.
Yeah, and then you have to work with the PI to say, hey, I was important for this, or give me a piece of it. But university owns it.
Okay.
Yep. Next one's from Sophie. I have a question about HIV/AIDS. You always say that viruses have a tendency to get less virulent because it lets the host transmit it to more people. Could the reason for the prolonged life of HIV patients be due to the virus that is adapting to us, or is it just the antivirals? All right, so there are two separate things here. We think that viruses should evolve to be coexisting with their hosts and not wiping them out.
In many cases.
But there are obviously exceptions. There are viruses that wipe you out. But those tend to be accidental, dead-end infections like Ebola and Marburg. But the virus infections that have been with us for years are pretty well adapted, and they don't kill us all, or if they do kill people, they don't kill all of us, and they take time so that the virus can spread. So the idea of a virus evolving towards virulence doesn't make a lot of sense to me. unless that is a side effect of it becoming more easily spread.
Right.
So viruses want to be transmitted. They evolve towards the greatest transmission. And if it happens that they become virulent at the same time, so be it, right? Now,
Right.
prolonged life of HIV patients, well, it depends what you're talking about. Yes, the triple therapy with antivirals allows people to live a long time. But remember, the course of AIDS, even untreated, it can be up to 10 years long. There's initial acute infection, and then you reach a point where you exist many years, and then at the end of that time, you lose a lot of T cells, and then you have opportunistic infections. So it depends what you're talking about. But it's not evolution of the virus towards less virulent. It's simply because if you're on drugs, of course, you suppress replication, or if you're not, that's the natural course of infection.
Yeah, usually when we talk about evolution of viruses to be less virulent, we're talking, long periods of time.
Right.
Many generations of hosts and viruses. Though it is also true that HIV evolves very fast in an individual. I don't think we're really seeing attenuation during that process.
No. It does
By the
evolve.
way,
It does evolve to evade all the antibodies you make, for example.
I heard an NPR report, I guess it was last night on the way home, that interviewed a couple who had been living with AIDS for 30 years. So they were infected in the 80s, and they've been through really all of the different kinds of drug treatments and a lot of horror, but they've managed to keep up with it, and they've been alive with this for 30 years. It's really pretty amazing.
They're on antivirals, I presume,
60s.
right?
Oh yeah, and they're in their 60s now. Actually, one other thing I'd like to just mention in this regard is that, as we've talked about before on TWiV, a large fraction of the ape immunodeficiency viruses don't cause disease in their hosts.
That's right. Yeah.
In the chimps, apparently they do, though it was originally not thought that they did. But in other apes, they don't. So that could be a situation where those viruses have had a longer time to evolve. I'm making this up, right? But where those viruses have had a much longer time to evolve in those hosts and have come to something more like an equilibrium.
That could be in 1,000 or a few thousand years. In people, It would be similar if we're around. Speaking of apes, did you see the Neanderthal sequence came out?
I heard that report on NPR as well and saw it in the paper today, but I haven't looked any further into it than that. But boy, were the scientists excited about that one. The NPR report was great.
And did you hear what is suggested by the sequence?
No. Oh, yes, that humans or human-like and Neanderthal actually mated.
Yeah.
Yeah. Fascinating.
So that's very interesting.
Great stuff.
She also says, furthermore, could it be possible to find the mechanism that controls whether it is HIV or AIDS you have? I'm thinking of something like bacteriophage lambda with Crow and C1, CI or C1? I forgot. C1. Or is it simply when a high enough number of T helper cells are infected, you will get AIDS?
It's the latter.
Yeah, it's the virus integrates into your cells, the genome integrates, and it just replicates continuously, produces viruses, and when enough T cells are killed, you get opportunistic infections, yeah.
HIV is the agent. AIDS is the disease that you get when the agent really takes over to the extent that you can no longer defend yourself.
So the virus doesn't directly kill you. It's the other infections that kill you that you can't fight off, basically. Okay, thanks, Sophie. Still love your podcast, she says. She's got me really worried that someday she's going to say, I don't love you anymore.
Nah, that won't happen.
I'm really worried about that, Sophie. Thanks.
Sophie, Vincent needs some reassurance. Ken writes, while discussing the evolution of H1N1 2009 pandemic influenza virus, Alan dropped the line, be a virus, see the world, which, of course, is a Far Side caption. When teaching introductory introductory microbiology courses, I used to disclose to the students at the beginning of the virology lectures that I understood that they had, coming into the course, very little knowledge of virus biology. I understood this because one can get a sense of what Americans learn in high school and introductory college biology courses by scanning the far side. A high proportion of Gary Larson's humor played on shared popular knowledge of biology. The fact is, and I know this because my beloved partner bought me the complete collection, that in 15 years of daily panels, there are exactly 2 that make any reference to viruses, and neither addresses virus biology. I'd actually like to see those 2 panels. I've seen most of Larson's work, but I can't think of those 2 panels. So, Ken, send us those panels. I no longer do that bit because my current students don't know who Gary Larson is. What a shame. And more of my students will already know something about the biology of viruses because they're growing up around people who listen to TWiV. All right.
It's beautiful, isn't it?
Excellent. Yeah, that's very nice.
Hey, that could be our episode title, Be a Virus, See the World.
Sure, absolutely.
And I know there was
if Ken gets on. Oh no, Ken's not going to be able to listen to this in time. Maybe I wonder if we could find that Gary Larson thing. It'd be nice to post that as the picture.
Yeah, maybe we could find. I have one here on my desk. It's at a Larson. It's a picture of a couple of buildings, and there are people in white coats walking around. And there's a broken jar on the sidewalk. They're all looking at it, and the sign says on the building, "Center for the Study of Viral Pathology." And there's a guy looking out of the second floor window, and he's going, "Uh-oh."
Oh, maybe that's one of them.
That is one of them.
Use that. That's great.
But I don't know what the Be a Virus, See the World. I've seen that, and I don't know where it is.
We can probably, if Alan were here, he'd already have it.
Yeah, he would.
Right? He would have it Googled. But we can probably find that.
We'll find it. All right. That's cool, Ken. Thank you. Duncan writes, "This is the second time that I have emailed you, and I have no desire to be considered a stalker, but I thought that I should let you know about the impact that TWiV and TWIP is having on at least one of your listeners. You can write us all you want. Don't worry about it. Last time I emailed you, I told you that I was planning on leaving my IT job to attend university and study for a degree in medical laboratory science. That was probably a year ago, and I still haven't left my job. Why, I hear you say. Well, I'm writing to tell you that it's pretty much your fault."
This is a great letter. I love this.
You see, originally I wasn't even considering getting into research, and that is why I was looking at the lab science degree. After a year or so of listening to your wonderful podcasts, I discovered that my passion for microbiology runs much deeper than I had previously thought. After much thought, I decided that if I was going to follow my dreams, I should follow them all the way, and I am now going to be studying for a Bachelor of Biomedical Science, majoring in genetics and molecular pathology. I'm not entirely sure what I will do after I've completed my degree, but I have an open mind and deep fascination in viruses, parasites, bacteria, genetics, disease, and what makes life tick at the cellular level. I may go on to do a PhD. I might even decide that after all that time at university, I'm satisfied with my level of knowledge and go back to IT, but I very much doubt it. Whatever happens, I thought that you would like to know that you guys played and are still playing a major part in helping me to achieve my dreams with your inspirational shows. Thank you very much, and keep on what you're doing. Keep on doing what you're doing. That's great. That's Duncan from New Zealand. That's amazing. That's great.
Yeah, that's amazing.
I love it. I don't think we have to say anything else, right?
No. Thank you, Duncan, very much. And good luck. Keep us posted. Bell writes, "I am appalled at the lack of sanitation in manicure salons." I'm glad after reading this that I don't do this. Even those salons that claim to disinfect and sterilize seem to cut corners. Has any study been made to determine viral transmission in these establishments? Many women have cuticles cut and pushed back, basically a surgical procedure done under non-sterile conditions. In addition to well-known viruses, other retroviruses, including XMRV, might possibly be transmitted. There have been clusters of diseases in many neighborhoods of different illnesses. It would be tantalizing to explore the unsanitary conditions of these salons as a source of infections. This rings a bell. I think there's been something like this done. Years ago, it was said that upper middle class women might have higher incidences of breast cancer I'm not sure of the status of this statistic. Has anyone ever questioned these women about their attendance at manicure salons, beauty salons? If you want to satisfy your curiosity of how unsanitary these establishments are, visit one and observe. Instruments are either not sterilized, sterilized inadequately, or reused from person to person. One establishment had their instruments in some kind of sandy material, which they heated to 135 degrees Fahrenheit. They were proud to show this to me. Others have instruments soaking in liquids. Nail shavings are left on paper towels from person to person. I'm sorry if this email seems eccentric, but it's very disturbing to see the mess that goes on in most manicure salons. I do have some substantial medical knowledge. In addition, my nails look very ungroomed. Sincerely, Belle. I think she's serious.
Well, there actually have been quite a few studies
Uh-huh.
done on this. And just to pick one out from 2004, role of beauty treatment in the spread of parenterally transmitted hepatitis viruses in Italy.
Wow.
So they conclude certain beauty treatments play an important role in the spread of HBV and HCV infections in Italy. They were manicure salons, but also barbershop shaving for hepatitis B virus. So, that still happens in some countries. You go into a barbershop and they use a long razor to shave you. Of course, that causes little microcuts.
Right.
And they say it's a good way to get hepatitis B. They say tattooing, piercing, manicure, and barbershop shaving they looked at. So there are quite a few. If you search PubMed for manicure and virus, you will get, let's see how many papers. 7 papers. Just with those search terms.
All right.
Turkish population, Italians. We got one on herpes simplex virus. So, yes, you're absolutely right. If the instruments are not properly sterilized, they can potentially transmit virus infections. Yep. Good point. We've never really addressed that before, have we?
Good point.
Yep. All right. Thanks, Belle. That's great. Let's see. What do we got here?
Matthew.
I just discovered TWiV and have been devouring episodes. The one on the 7 types of virus genome was the best one yet. I will have many more questions to come, but for now I have a somewhat practical question as I'm currently recovering from a recurrence of herpes simplex in my cornea. It's interesting to think that I'm actually looking through herpes simplex viruses as I type this. You are hooked on viruses, Matthew.
That's serious.
It's okay. I have acyclovir on the case. What I'd like to know is there any truth in the idea that eating foods rich in arginine can trigger a recurrence of herpes? The idea is that eating more lysine-rich foods can prevent recurrences by blocking absorption of arginine. Thanks for doing TWiV. I love the episodes with you and Dick talking in technical detail. I think that viruses are beautiful, enigmatic things. Surely they hold clues to some of the profound mysteries of cellular life.
Well, he obviously gets it. That's good.
So your friend had some good comments on this one.
Right.
David Bloom, who is a herpes expert at Gainesville, right?
Right next door to me.
Want to read that?
Sure. It says, when HSV productively replicates...
That's the wrong one.
That's the wrong one.
You
Okay,
have to
so
go back
hang on.
up. Yeah.
I got it right here because I got his email in front of me. So there is no compelling clinical data supporting the assertion that arginine triggers recurrence. That being said, there are a large number of people who experience frequent recurrences that have documented that changing their diets to avoiding foods high in arginine really helps to control the frequency and severity of recurrences. As you can imagine, though, it is hard to link a lot of these dietary changes and their effect on recurrences directly to arginine content. So for the lysine part of the story, here there have actually been a number of in vitro, in vivo, and clinical trials looking at the efficacy of lysine in blocking HSV replication and recurrence. High concentrations of lysine can reduce acute replicative yields of HSV in culture, but do not block reactivation of HSV in explant models. In clinical studies, there is no consensus. There seems to be a lot of variability in its effectiveness from person to person, so while a lot of people swear by it, it is probably at best a cofactor that can help prevent severity/reactivation in some people. He gives a link to a recent review with some good references. So, it's controversial, right? In the case of arginine, it's anecdotal. In the case of lysine, there's some laboratory data, but the clinical data aren't good, and you have people that swear by it. So it's far from proven.
What foods are rich in arginine
Gee, I don't know. You know,
or lysine?
this I don't know.
I
Dick
don't,
could probably tell us, but he's in
yeah,
Seattle.
or even Alan could.
Alan,
Let's,
well,
let's
Alan would have it all Googled by now.
let's see. Foods rich in
Okay.
lysine. Let's see what we get. Herpes diet and foods information. Can you believe it?
Yeah, of course.
These include red meat. What else? Wait a minute. Now I'm stuck on this page.
I get this. Oh, yeah, I get the same thing.
Red
Diet,
meat, pork,
herpes
poultry,
diet foods information.
Parmesan cheese. Cool. I like Parmesan cheese. So there are specific foods rich in them. I don't see that it would hurt to eat those foods, right? A bit
Oh,
of red meat. I mean, that's just what many people eat anyway, right?
right.
Foods rich in protein. Well, that because lysine is an amino acid, that doesn't
Sure.
help. I don't think it's fenugreek seed. Brewer's yeast. Hey, just drink beer.
All right.
There you go.
Drink a lot of beer.
Drink beer.
There you go.
Thanks,
Then you don't
Matt.
care if you got herpes anyway.
Let's see here. What should we have? Should we finish these up? We're almost done.
Hey, you're the boss. I'm ready. I'm on a roll here. We're almost done.
All right,
For
we're
sure.
almost done. Laura writes, is it my turn or yours?
Gee, I don't know. I've lost it. I think it's my turn.
All right, go for Laura.
Okay.
Yeah.
Laura writes, Dear Vince, Dick, Alan, and Rich, thank you so much for your wonderful podcasts, as both TWiV and TWIP are my favorites. They frequently make me laugh out loud, getting me strange looks on public transport and at the gym. I teach high school biology in Hong Kong, and I'm currently doing a distance learning MSc in infectious diseases, so all your info is invaluable. I'm writing to recommend a pick of the week, which is Arno Carlin's Plague Process, which Plague's Progress, which is both informative and very easy to read stroll through the history of disease. If any of you are ever over in Hong Kong and you're at a loss for what to do, send me an email, and I will happily show you around and say thank you. Regards, Laura. Well, that's a nice invitation. I don't know this book. Do you?
No. I'll check it out for
Have a
sure.
look at it.
See
Great.
how
Thank
it
you
is.
very much.
I've never been to Hong Kong. Have you?
Neither have I. Nope.
All right. Thanks a lot, Laura. That's great. James writes, I was flicking through my RSS feeds when I came across this story about the New Zealand Blood Service moving to stop people with CFS from chronic fatigue syndrome from donating blood. This seems to be a precautionary measure until the science becomes settled, as it is remarked in the story that the science is shaky at the moment. So this is an article from a New Zealand website. Chronic fatigue donors face rejection. Yeah, they're not being allowed to give blood, which seems like a reasonable restriction, given that there may be XMRV in their blood, right? So
Yeah.
I don't think that's an issue. It's not prejudicial. It's just common sense.
Right.
And a quick question. If H1N1 swine origin influenza is found to displace the H3N2 seasonal strain, would you expect the WHO to switch their suggestion to a bivalent vaccine and thus instantly increase the amount of seasonal flu vaccine that could be produced by a third? Just an interesting public health logistics thought I had. I don't quite understand that. So this season's over in the northern hemisphere. I guess down in New Zealand, the winter is just beginning. And so you guys are going to be using the vaccine that we've had all along, which is a seasonal vaccine, which is 3 components, and then the pandemic vaccine. So would we switch? The only time you will switch is for the next season. You don't switch in the middle of a season.
Right.
So for up here, the winter, that is this winter coming up in the northern hemisphere, the vaccine has already been decided. It will be a bivalent vaccine consisting of H3N2 and swine origin H1N1, which is basically
doesn't
now.
it have a B strain in it as well?
Sorry, it's got a B strain as well. It's trivalent.
Okay.
So the swine origin is now a seasonal strain starting in the fall here.
Right.
So I don't know about making a third more vaccine. Do you understand that?
No, I think the idea is it's almost as if there was a packaging issue here or something, and there's not.
No.
So if you were to reduce if you were reduce the viruses from uh, 3 to 2, you'd still have the same amount of each of the individual viruses. So it's not gonna change the quantity at all.
Right.
So and trivalent is usually the deal, right? You do two A strains and a B strain, the most
Right.
logical
That's
A
right.
strains and a B strain.
Yeah. So in fact, this past season it was a trivalent and a monovalent because
Right,
the
because the
pandemic was
pandemic.
given separately.
Right,
They never tested a tetravalent, so they couldn't just
right.
introduce that. Right.
So
Okay. You're next.
Stephen writes, I was listening to your story about researchers who bought an assortment of groceries in Marseille for testing. I wonder if they bought any koala brand tea. This is important because the koala tea of Marseille is not strained. Okay.
I never
Hey,
heard of koala
no,
tea of Marseille.
no,
Have you? Let's look
no,
it
I
up.
have not.
Let's see. Koala tea of Marseille. Maybe it's a joke.
I think so, but I don't get it.
The Koala Tea Company, which is Australian herbal teas. That's certainly not Marseille.
Okay.
Um,
That's a pun on, I forgot that the quality of mercy is not strained, right? It's a Shakespearean line. Okay.
oh, you got it.
A bunch of science geeks. Oh, I cheated. I got it on the internet here. Okay.
Oh, now, so
The
that's why he has a little.
quality of Marseille is not strained. I knew it had a ring to it, but I didn't quite get it. Okay.
Nice. That's why
Yes.
he has a little smiley at the end
Yes,
there,
we
right?
just had our legs pulled. Thank you, Stephen.
Thanks, Stephen. But yeah, actually, it probably does. Well, it might not have that virus, that plant virus, because it's a pepper. It's a pepper virus, chilies and peppers. But it's a funny joke. I got it. I hadn't gotten it. Good job, Rich. All right,
I'll get
let's.
better at this Google thing, Alan can do it. I can do it too.
Good. That's great. That was a nice bunch of email.
Yeah, that was great. Good fun.
By the way, everybody, I now post the complete text of all the emails at twiv.tv/letters. So you can read them if you want. So that brings us to our science picks of the week. And Rich, you have a neat one this week.
Yeah, okay. So my son works for Google, and he
Oh,
sent me this
cool.
thing. Yeah, it's very cool.
You know, Saul's son works for Google too.
Is that right?
Yeah, out in San
Where?
Antonio.
In San Antonio. Okay, so
Yeah.
my son's in Redmond, Washington.
Mm-hmm.
So he sent me this link to a Google Chrome browser speed test. And I've added a couple of links here. One's to the Google blog, and another is to the YouTube of the same thing. The blog may go away eventually, I suppose, but the YouTube thing will be there forever. And it's basically an advertisement, okay? But it's a very clever ad. It's what I would call not pseudoscience, but faux science, where the theme is that they're testing the speed of the Google browser against various other phenomena like sound or lightning or something like that. So they set up these sort of Rube Goldberg tests that I can't even describe, and set them off so that they set off the test and a Google search at the same time. And of course, the browser always wins. And they're shot in slow motion and stuff. And one of them's a potato gun that's an absolute riot. So I suggest you just have a look at this, and you'll find it entertaining. And I must say, of course, with your son working for Google, you got to start using all the Google products. And he told me I had to use the Google Chrome browser. I must say it is lickety split.
It's very nice, yeah.
It is noticeably faster than any other browser I've used.
Now, this is great. It's kind of pseudoscience, but
Yeah,
it's a
but
lot
it's
of fun.
I mean, they know it's pseudoscience. It's
Yeah.
a joke. Okay, but
They're beautiful. They're beautiful movies. It's just amazing.
They are, and it's the composition, all of the marketing is very it's very artistically done. There's also another link here that I put on how it was made.
The making of.
The making of.
That's
so
great.
they interview the people who did it. So that's good.
Cool. Well, my pick this week is a book, and I'm reviewing this book for a journal. So I'm almost done with it. It's called Inside the Outbreaks by Mark Pendergrast. This is it's not really a history, although pretends to be, but it's a recounting of the Epidemic Intelligence Service, which is a branch of the Centers for Disease Control. And these are the guys, you know, what's that clip from the Ghostbusters? Who are you going to call?
Yeah.
Right. So if you got a disease outbreak, who are you going to call? EIS, basically.
Right. These are the They're the SWAT team.
They're the SWAT team. They were founded in 1951. It's a very interesting story, actually.
That sounds interesting.
They were founded by Alexander Langmuir, and he said in 1951, you know, we're susceptible to bioterrorism, so we should form a branch of the CDC, which would be like a quick response team. And it so happens that in 1951, we're in Korea for the Korean War, and 25,000 American soldiers got Korean hemorrhagic fever. So he said, look, see, this is why we have to have this branch. So the government gave money, and they started this branch. And over the years... So are those
the guys who, like, go out to Four Corners when the hantavirus thing...
Yeah, wherever there's an outbreak, they send... It doesn't have to be infection. It could be something else. You know, recently in the pig slaughtering factories in the Midwest, there was an outbreak of a neurological disease, which turned out to be from inhaling the myelin in the... when they were taking apart the pig neurological tissues. So they'll investigate anything. And so this is a collection of stories of different outbreaks over the years. They're very short. Each one is about a page or two. And they cover a couple of different phases in the history of this. You know, smallpox outbreaks, polio associated with the vaccine. So, you know, if you like reading these little vignettes, you'll like it.
That sounds great. So this is newly published, and you're reviewing it? Is that the idea?
Yeah, it just came out last month, and I'm reviewing it. I'm going to write a review soon.
Cool.
Now, the cool thing is how many people were agents of the EIS. So the EIS, you spend just a couple of years there, and then you move on. Mm-hmm. D.A. Henderson was an EIS officer, the guy who architected the eradication of smallpox.
Right.
Bill Fogarty, who many people may know, was at NIH for many years.
Right. Familiar name. Yes, right.
Neil Nathanson, who is a polio guy. He's retired now, but he's at UPenn. And one more, I'm going to forget his name now. Darn, let me look it up. I think now he's Lawrence Altman, who is a science writer for the New York Times.
Very interesting.
So Lawrence Altman was a journalist who went to medical school, but he didn't really want to be a doctor. So he spent a few years, and he was actually the first editor of the Morbidity and Mortality Weekly Report. So anyway, it's a cool book. The cover is fabulous. If you go on Amazon and look it up, it's got a great cover. Right. And you'll love it.
Great.
Inside the Outbreaks.
I'll have a look.
All right. Well, that'll do it for another TWiV. As always, if you're a new listener, think about subscribing. You can go to iTunes or the Zune Marketplace. And in iTunes, if you do subscribe, please leave some comments. It always helps us stay on the front page. I'm really happy to announce a new way you can listen to TWiV, and that's called Stitcher, Stitcher.com. And this is so you can listen to TWiV on your mobile device. Now, up till now, you could listen on your iPhone. But now if you have a BlackBerry, a Palm, or anything running Google Android, now you can listen to TWiV on them. All you do is you download Stitcher, which is an app. It's a free app. And then search for TWiV, and you'll find it. We actually have a landing page, and I'll put the link in the show notes. But then you can listen to TWiV on the go. It doesn't download it. It just streams it to your device.
Excellent.
So Stitcher.com, check that out. We appreciate having a partnership with them. We're also part of sciencepodcasters.org, promednetwork.com, and microbeworld.org, websites where you can find a lot of great science content. As always, send your questions and comments to TWiV at twiv.tv. Rich, thanks for doing it again.
Hey, I love it. It's good fun.
Yeah, I always enjoy it. A lot of fun today doing email.
Yeah.
Rich is at the University of Florida, which we will be at in a couple of short weeks doing TWiV. Look forward to that.
Great.
You've been listening to This Week in Virology. Thanks for joining us, everyone. We'll be back next week. Another TWiV is viral.